Hello, and welcome to the Power of Omics. My name is Jenny Ho, and I'm an EMEA Sales Support Specialist for Proteomics at Thermo Fisher Scientific, and I will be your moderator for today's event. Today's webinar session is part of a special event we are running this year in advance of the International HUPO Connect meeting outlining the power of proteomics in key application areas. This event will discuss how scientists are harnessing the power of proteomics to advance scientific research. Over the 3 days, you will hear first from our plenary speaker in the opening session, followed by selected scientists in the second daily session, who will provide an overview of the current state of research in their individual fields. The third session will feature a roundtable discussion led by a prominent researcher, providing a look towards the future and giving you the opportunity to join in with what I hope will be a very interactive and interesting discussion. Day 1 of the event focused on tackling virus research. Day 2 of the event focused on proteomics and cancer. Today is day 3, and the focus of today's session is Go Beyond Unleashing the Power of Proteomics. In the last decade, proteomics has become a widely adopted technique, often multidisciplinary, requiring collaborations with biologists, protein chemists, mass spectrometrists, and bioinformaticians, delivering new innovation and diverse applications and workflows to understand biology. Modern proteomic solutions rely on scientific community pushing the limits of mass spectrometry technology and going beyond mass spectrometry with complementary developments in sample preparation, fit-for-purpose reagents, methodologies, and more. This session highlights several of these collaborations, including single-cell proteomics, native MS for structural analysis, multiplex proteomics, and artificial intelligence that enables mass analysis of proteins from any organism significantly faster than before and almost error-free. Continuing with the theme of community collaboration, we will also feature a roundtable session that will discuss future needs, both technology, applications, and the need for standards that will deliver the promise of proteomics for human health. As we go through the program, please use the Q&A function to type in your questions, and we will try to address as many of these after each presentation. So without further any delay, I'd like to introduce our plenary speaker, Professor Neil Kelleher from Northwestern University. So over to you, Professor Kelleher.
Great. Thanks for having me today and to talk at this Thermo-sponsored event and to connect with you, even from afar and through recorded means, on measuring protein-level biology, one ion at a time. So I'm happy to be here and present on a wide range of topics, and let's dive right in. Our summary here is, I thought I'd warm us up with what's on the minds of many in a worldwide pandemic, and that's just a little bit of science around SARS-CoV-2 and COVID-19, and then move into how some of the data I'll present was created, and that's this really exciting development of individual ion-MS. I'll refer to it as I2MS for short, and we'll take a veritable romp through a set of topics, including antibodies, and getting some world records on resolving power set with this new technology, and using it to deconvolve protein complexity. And I'll show some native and top-down mass spectrometry on VLPs and other topics of the day. At the very end, I'll share with you some new advances and a new stream of separations and how to prepare samples for things like I2MS in a really new way. So let's dive right in. And, many of you may know about all of the vaccine development being sponsored by Warp Speed here in the United States. And Novavax has received the majority of funds here, the largest amount of the funds from one sole company. And that's about $2 billion in total. And that's been pumped into Novavax, which is a protein-based vaccine, a nanoparticle that presents the spike protein of the virus in a way that mimics the actual surface of virions. And it's been the focus of a lot of press, and certainly $2 billion. So that's 1.6 billion US from the federal government of the United States, and then another about 400 million from a not-for-profit entity called CEPI, And, you know, what it's taking is an approach to vaccine development that's focused on this glycoprotein, the spike protein of SARS-CoV-2. I'm not going to belabor the introduction of all the virology and other things. This isn't a COVID-19 talk, but I will say that both in Europe and the United States, this podcast called TWIV, This Week in Virology, it's hosted by this fellow Vincent Racaniello and embraces several guests from European countries. And it's just been a tremendous growth potential to learn about immunology and virology. And so that's what I've been doing during the pandemic. Let's focus on this glycoprotein, the spike protein from SARS-CoV-2, and let's look at its structure articulated with all the glycans. And that's what you see in magenta here on this slide, is the glycans, several dozen N-linked glycosites are on this protein. And, you know, the speed of this research, our response to the pandemic as scientists and biotechnologists has been overwhelming. And the pace of this is just unprecedented. So here's just a small sliver of this to get us warmed up into our talk there this afternoon in Europe. And I just draw your attention to this structure of a neutralizing antibody with the spike protein. And I'm going to just focus in down on panel D here in the lower left. And so let's zoom in on that. And what it is, it's actually mediated by some of those glycans. So on the spike protein of SARS-CoV-2, there is an N-linked glycan on asparagine 343, highlighted here. And the neutralizing antibody has both light chain and heavy chain recognizing the spike protein through that glycan. So that's just a little bit of science to tee up just that the protein diversity in that world is critical to understand and to better measure. And so the pandemic has just laid this bare, and I hope that you and yours have been safe and that you too have been energized to try and improve the world through science. And so let's get into things you're more used to in this kind of forum, and that's a mass spectrum of the spike protein. So this is the receptor binding domain, the part of the spike protein that is recognized by ACE2 receptor on the surface of human cells. And so this receptor binding domain, the part of the spike protein, is also heavily glycosylated. And here I just show you a spectrum of the glycoproteoform profile in the 30 to 35 kilodalton range. Now, the underlying protein is itself 26 kilodaltons, just over. And you can immediately see that the shift in mass is due to the glycans, about 5.5 kilodaltons of glycans. And you see that profile. And so it's trivial now for us to get this mass spectrum. And I'll tell you why that is and how that is in just a moment. And so we can do this in denaturing mode or do this in native mode. And here in native mode, we see that this construct forms a dimer and a trimer like the natural spike protein itself. And we can get isotopic resolution on all these different forms, even up to 100 kilodalton at low intensity, and get these glycoproteoform profiles. I mean, this is now trivial to do. And so to get this spectrum and this information before the advent of I2MS versus after is like night and day. And let me go on to tell you some more about that. And so I2MS is a new technology that leverages a non-incremental improvement in the past couple of years that we've been working on. And it's been now, there's 6 publications with 2 more coming. So it's a veritable storm of papers on this. And let me highlight that for you. And let me also tell you the story about how this came to be. And it basically uses an Orbitrap to assign the charge on each and every ion that is passes through the hardware measurement and software processing that we've set up. And this has been a collaboration with the group of Mike Senko at Thermo Fisher Scientific in San Jose, California. And his team and mine, we assembled ourselves in October of 2017, and then proceeded down this very fast train of development. And that's the story I wish to tell you today, and to acknowledge the whole team that pulled this off. And I really believe this to be, as I said, a non-incremental gain. And so why do I believe that? What is it about? These are the things I'll answer, and I'll actually move through the talk fairly quickly. I appreciate that it's a virtual event, and but let me try to engage you as best I can, and then answer some Q&A in a few minutes. So charge detection mass spectrometry is not new, and it has used single ions in the past, all the way back to the late 90s with a fellow named Henry Benner. More recently, Evan Williams and Martin Gerald in particular have advanced an ion trap format where they get very good charge assignment accuracy on one ion, or maybe up to 10 ions at a time. But the positioning of that ion on the mass spectrum is not great. Of course, an Orbitrap has great frequency accuracy, but before this effort, it was not possible to assign the exact charge on an individual ion. Now it is. And we can then take advantage of the fantastic frequency accuracy of the Orbitrap. And as we've shown, we can now move into the thousands of ions per shot into the Orbitrap and do this it's kind of paradoxical, but you could understand it as a multiplexed individual ion mass spectrometry approach. So we can do what I'll show you on thousands of ions at a time. In this paper, referenced at the bottom left of this slide, we were doing it on 110 ions at a time on average. And basically, you bring a population of 110 ions into an ion trap, like the Orbitrap, and you seek to use the intensity to assign what is the charge. So in this case, you're looking at, on the left-hand side of this slide, the charges of myoglobin, charge states, ions of myoglobin, and you're looking at a histogram of intensities for the different charge states. And you can see they're all over the place. And using intensity alone is fraught with challenges and problems. And so we've gotten around that. And on the right-hand side of the slide, what we use is the central limit theorem to converge on the correct answer. And this is, in this paper, it's quoted at 96% of the time, but we push that now also to approach 99% of the time, the accuracy of the charge assignment. And let me tell you how we've done that. There's all sorts of challenges in this business, like ions dying during detection, or two ion events that happen to overlap, etc. So how do you make this a robust process? And here's an example of now connecting with some of the raw data for that underlies I2MS. So these are 4 ions of the 20+ charge state of myoglobin. And the goal of the exercise is to say that each one is 20+, right? These are isotopic peaks that you're looking at. So A and B are isotopic peaks for well-behaved ions. And why well-behaved, what I mean to say is that over a 2-second detection period, as they're spinning through the Orbitrap, you can integrate the current that is induced by those ions as they fly through. and fly around the center electrode. And so as you integrate the accrued current over time, you can see that over a 2-second period in the graph in the upper right, that you get more and more integrated signal. And it's actually the slope of this ion that we have locked in on and said that slope, and we call this a STORI plot, so it's a STORI slope, and that's all published in the paper and referenced in the lower left of this slide. But that slope defines the charge state, right? So if that's a 20 plus ion, then a 21 plus would have a slightly higher slope and a 19 plus a slightly lower slope. Now, ions C and D are fragmentation events, and so they aren't well behaved, but we can still recover them as 20 plus charges. And that's because we're using the slope, not the intensity. And that's how the core of the method works. Again, this is published now. This was the cover article from Nature Methods in April of this year. And it is diagrammed in this slide here. And let me now quickly take you through again the process. And now we're going to multiplex it. So here comes 110 ions on average shot into the Orbitrap. And right there, let me just stop and mention that we actually have to detune the instrument to make sure that just a few ions are entering at any given time. So the one way to detune is only inject ions for a very short burst, so sub-millisecond bursts, or you can use very dilute solutions. And I'll only make this point now, but that, we've, instead of using low micromolar concentrated samples, we now use concentrations of low nanomolar. And that, too, is a way to make sure that 100 or 1,000 ions per shot are going in. OK, with that digression, let me reconnect with the slide here and note that we shot in about 110 ions, and you're looking at the frequency of those in the upper left. And then you go through the steps of the process, right, which for every individual ion, we create this STORI plot, as I showed you. And we anchor on the slope of that plot to determine the charge on that ion. And we refer to a calibration. I'll mention the calibration to you shortly and show you that it's quite stable over time, over months, periods of time. And then we can produce a true mass spectrum, a depiction of those ions in the mass domain. Because we know the charge state and the m/z, or frequency, we can create a true mass spectrum. And that's the breakthrough. So the STORI slope here, we actually have to calibrate it. So here you have both in denatured mode in red and in native mode in blue. And so we've used a Q-exactive plus, or now an HF, and a Q-exactive UHMR, which we co-developed with Thermo about half a decade ago. And we have got the slopes now that we can now use as reference. So the STORI slope is now the y-axis, and the charge is on the z-axis. And using known calibrants, we now get a STORI plot. We get a slope for an individual ion. We then refer to this plot and then determine the charge. Also co-adding many common ions at a time, and this improves our charge assignment accuracy. So this is stable amazingly over weeks and months. It doesn't require major hardware changes and just a bit of software to be able to implement this. And this is being increasingly supported by Thermo and beta tested, et cetera. So it's in the pipeline. Moving forward, this was highlighted in a CNE News article. So after calibration, we can now use it. And the approach is benchmarked here on a standard monoclonal antibody, and not even one that's clean. And what you've got here is just a reduced monoclonal antibody into its light and heavy chains. And in normal standard MS, you have the left-hand slide here. The data set shows the rising baseline that is standard in the field. And this is because of a lot of glycosylations and a lot of co-occurrence of the ions. And you see that rise baseline. That is completely different when you go to I² MS. And on the right-hand side, you see this plot where the light chain and the heavy chain in the lower right here have a very different appearance. And there are some guanidine adducts at plus 60 Daltons also, making this a more complicated spectrum. But it's readily read out, and the baseline is completely flat. It's a very different sort of world than standard or ensemble MS when you go to I2 MS. Now, we have used the technology for a couple of years now, and it's like any single molecule approach. And here we have the positioning of the ions in frequency is also advantaged. So when you have a single ion spinning inside the instrument, its frequency precision can be asserted with much better accuracy because you know it's a single ion. So you can position it accurately. And then you acquire many thousands of such events, and you can now build up a isotopic distribution easily at what you might call a super resolution. And so this is beyond the instrument settings that you would normally expect a resolution of X. You get 20X gain. And this was reported back in 2019 on a monoclonal. We've since moved beyond that. And with I2MS, we have a world record now, not once, but twice on this slide. Let me explain it to you. So using two tetrameric homotetramer complexes, pyruvate kinase and beta-galactosidase. One on the left is PK, pyruvate kinase, at about 232 kilodaltons. And the other one is over 466 kilodaltons on the right. In both cases, we can get isotopic resolution on an Orbitrap. I mean, that's just amazing. And we do it by what I just said and showed in that prior article. So when you resolve things isotopically, you can make sure that you're not shifting the mass by the underlying species because you've resolved them. And so pyruvate kinase has some mild differences here, including a truncation of one of the subunits, one of the 4 subunits here on the lower left. That's this peak at around 229,500. For beta-galactosidase, we have a major shift in mass from that expected. And we go on in this paper to describe why that might be. But there's no question that it's unit resolution, as showed in the inset on the lower right. So that just it shows one or more advantage of I2MS. And let's continue on. So here's another depiction of this where the experiment we did here, which is in the paper in this article mentioned before, we took the entire whole cell extract from HEC293 cells and isolated the 5 to 30 kilodalton chunk of the proteome, so the entire mixture there. We then cleaned it up in the ways we normally do prior to LCMS, but didn't do LC. We simply injected it into the instrument and did an ensemble measurement, and you can imagine the result of this, right? Which is, we call this affectionately the hump of death. That is way too many charge states for way too many proteoforms all overlapping. So it's unintelligible. You can't determine masses. However, if you do the exact same experiment and acquire data, I2MS data on the right-hand side, remember that baseline I was describing? You can see the flatness of the baseline, And now we've detected 1,000 different masses for proteoforms. And if we now refer to the LCMS experiment and the set of proteoforms and their proteoform records in a database, the intact mass values of them, we can begin to assign some of the 1,000 masses, and we did that for about half of them here, using the intact mass as a basis to assign a proteoform record number in a proteoform atlas that sits with the Consortium for Top-Down Proteomics on its website, which is tdprot.org. So that just shows you night and day, right? You've got left-hand ensemble measurements. You can't get any mass values out of it. And on the right-hand side, you get super resolution and clarity about the mass spectrum on a very complex mixture. So this is the basis for my statement about I2MS that we can approach any target in biology. Could be the spike protein from COVID-19 and SARS-CoV-2, heavily glycosylated, hundreds of kilodaltons, whatever the complexity of it is, this is the technology that will give you a mass distribution. Very exciting. And because many of you have tried Intax or top-down MS in various ways, but it's the sample prep, and there's this barrier to collecting first, can I see the proteoforms that are present in my sample? And now we can do that and get a mass distribution irrespective of the molecular complexity we're dealing with. And speaking of top-down, we've used the technology as well. This was the cover article for JPR, Journal of Proteome Research, from back in March. And what I'm going to show you is just a snippet of data here on triose phosphate isomerase, TPI. It's an enzyme central in glycolysis. And we'll focus on just the 39 plus charge state. So we isolated that and then fragmented it and compared with ensemble and with I2MS readouts. Let's look at the data. So on the left-hand side is the traditional ensemble readout, and on the right-hand side is MS, I2MS, or I call it tandem MS readout using individual ion mass spectrometry. That's the long form. Here is the readout. So if you look at the nature of these readouts of the tandem mass spectrum of the 39 plus of this protein TPI, it's about a 40 kilodalton protein. You just see the nature of the data type is completely different. On the left, you have the standard set of isotopic envelopes, each with a charge state that needs to be determined and a certain baseline appearance. On the right, you have a readout with a flat baseline and using the I2MS a platform. So what does it look like when you deconvolute or assign masses to all of these? Here are those data, and in one form we've used a graphical fragment map to show you where these fragment ions land. And I think the things to zoom in on are best described when I come back to this slide. Let me go ahead one slide and show you just in red are the ensemble, their standard approach, and in blue are the I2MS readouts of these fragment ions. You can see there's just a tremendous difference in the average mass of the fragment ions. That's shown on the left-hand side with the box and whisker plot. We're averaging somewhere around 20,000 daltons for the readout of these fragment ions of a 40 kilodalton protein versus something like 3 kilodaltons for the standard approach. On the right, you've got a colored depiction of the same thing. Now, if I go back, look at what we're looking at here. The standard MS is in the red fragment ions, and the I2MS readout is in blue. And when you look at the complementarity pairs that we're getting, so the red are the standard approach, and you get these small B ions in the top part of this graphical fragment map. But look at the blue ones. They're these massive Y ions, whereas you saw these smaller B ions. It's true in the middle of the protein. And then also on the C-terminus, it's the same thing, where you have small Y ions, standard approach, massive B ions, your B 330 ion, right? Or your Y 315, right? These are now trivial to read out. So I'm very excited about the potential for this in top-down. One other area of application are on VLPs, or virus-like particles, and assessing their composition and their cargo. And it's the same type of message, different application, and one that takes on particular significance during the COVID-19 era. And so here is a VLP. It's made up, it's a capsid from the MS2 virus, and it is sprayed and analyzed in standard ensemble MS and gave this data that you see in the lower left. It is unintelligible. You can't determine a mass value. All the charge states are overlapping. And it has heterogeneous oligonucleotide cargo, so a few kb of RNA and DNA are inside this capsid as it is expressed from E. coli. So on the right-hand side, you see the upper part of the right-hand is the I2MS data, and it's got a mass of 3.19 megadalton with a certain width to it, and that width is about 75 kilodaltons. And it's not due to the mass spectrometry, it's due to the biology. And I'll show you it's the length variation of the oligonucleotide cargo that underlies this. So here's all these charge states. We're working at between 131 and 140 plus charge states, and that's the answer of what was underlying this hump in the lower left. If you add up the cargo, the DNA and RNA cargo, and we sequenced the cargo, and so we know its length distributions, the DNA and the RNA, and you add it up, you get very close to that expected. And so we're reported this in the paper, and I'll show you one more example. There's actually a mini-me version of this, a mini MS2 virus-like particle. It's from a single point mutation of changing a single serine to a proline at position 37. This is collaborative with Professor Danielle Tillman-Ersick. And there are cryo-EM structures of these VLP particles. And from that, we know the number of oligomers and the diameter, right? So we've gone from 180 subunits on the wild type capsid here down to, and 27 nanometers, down to 60 subunits and 17 nanometers. So it's going to be a much lower mass. We're at 3.2 megadaltons on the far left. What are we on the right? And that's shown here along, and I'll just let this slide pause for a few seconds. These are the underlying sequences of the DNA and the RNA, and you can look at the length distributions and pause the talk if you like and work that out. It's also published. But it's the same basic deal, where there were, even at this lower mini-me version of this VLP, you couldn't assign the charge states with standard, but with I2MS, it's trivial to determine the mass distribution. And it weighed in at just under a megadalton, 0.99 megadalton, with a width about 32 kilodalton wide. And again, that's due to the heterogeneity of the oligonucleotide cargo. And the math there is in the center part of the slide. We're down to 60 to 70 plus charge states. So we're looking into fragmenting these and all sorts of fun with this new approach. Let me move on in the interest of time and come to the last part of my talk and focus on the sample handling problem, the front end problem of mass spectrometry. Now, we all know that this is a part of the secret sauce that is really critical to get right. If your sample is not clean and ready for mass spectrometry, you will get suboptimal data. We now can get you a mass distribution even under very dirty circumstances, But wouldn't it be great if we had a protein concentration device and a washing machine for automated buffer exchange that was applicable to up to a few tenths of percents of SDS and some of the gnarliest kinds of buffer components that we're used to working with. And we also need to have this work at the mid to low nanogram range. This is what sample stream represents and what I'm going to finish off telling you about. So the device itself is embedded in this on the CTC PAL3 robot, and so leverages all of the availability of that workflow. And then it's hanging here, this black 3 pumps. The actual sample stream channel is this Teflon white piece. And let me zoom in on the next slide of what happens inside the device. But it's this very efficient package that we've been working on for about 5 or 6 years. And it goes live here in the first quarter of 2021, a year that hope will bring vaccine, great weather, and more return to normalcy for more people all around the world. So for a sample stream itself, it's basically a protein concentration device and a washing at the same time. And so what it uses is counterflow after sample injection. The counterflow uses the chromatofocusing effect to line up those red balls. That's your analyte depicted here. And so that's the concentration effect. And then it also has this molecular weight cutoff membrane embedded. And because of that, you have this downward flow. And we're talking, you know, with the pumps that you saw, 100 microliter, 200, 400 microliters of stringent washing and buffer exchange while you're concentrating. So this is the magic of sample stream, is to be able to do that at the nanogram level. Now, when you have your sample processed and ready to, you've got to now get it into either a fraction. So the PAL3 robot is capable of fraction collection. Now you've got a concentrated sample in a different buffer with high recovery, or you can send it straight to the mass spectrometer. And this is what we're, of course, working on feverishly here during the pandemic. And what we do is we switch a valve on the PAL3 robot, and that diverts the flow into a robust source at, you know, anywhere between 1 and 50 microliters a minute into an Orbitrap mass spectrometer, and it gives a profile of elution just like a chromatography peak. And you know, with all the advances in mass spectrometry, even if this is a modest mixture, we will be able to do top-down, native top-down, or just get a mass distribution. And so here's a direct comparison of sample stream to LC, and the virtue of not having all the complications of LC are many-fold, and having the repetition rate of about, at the fastest we've gone is 15 seconds per sample, but to allow for very complicated samples and buffer exchange, this run was done of a monoclonal antibody in 2.5 minutes per sample versus 6-minute LC run times. And so it can go reasonably fast. It's at the nanogram level. This is 500 nanograms of Herceptin, and it gives the same kind of peak. And we've also switched between native mode and denatured mode, which also has advantages. So I think I'm going to move to exit and leave time for Q&A. I think that's really important. So I'm not going to belabor the denatured native mode switch for antibodies. We can also do enzymatic digestion in the channel or reduction of disulfides for antibodies, and or even bottom-up or PNGase digestion. So we've been demonstrating these things. These are the brains and workhorses that we've been collaborating with on sample stream. That's Phil Compton and Jared Drader. And of course, you know, I think in summary, you've just seen the central role of the Orbitrap and the continued massive innovation that's coming, you know, year in and year out, and making us able to do more with the same or less number of FTEs that work for us. I think this is a template for the future. And, you see if you agree with this statement about that first bullet point, is this a new paradigm for mass spectrometry? I think it is. And I think it lives up to that billing because of all the advantages I've described. Maybe even makes J.J. Thomson smile from his grave, where he, invented the Thomsonometer, and that means you always had a mass to charge. That's what we measure, right? But now we're producing an actual true mass spectrum. And so it's a mass spectrometer. It's not a Thomsonometer. So I think it is a sea change. And so I2MS, very quickly generates data, and now with the world record at 466 kilodaltons for isotopic resolution. Using it to profile glycoproteoforms on the spike protein of SARS-CoV-2 or any complicated molecular weight distribution. And we all know that that's happening in pharmaceuticals, biopharma, and advanced therapeutics, VLPs, and AAVs. They're just prevalent. And so this technology, and with an approach to solve the front end problem and really make what used to take PhDs able to be done with more junior staff and at lower price point per measurement. And so economies of scale will be coming. And with that, I think I'll just thank Danielle, who's been a spectacular collaborator on the VLPs and the virus-like particles with Bon and Kwagu, and the whole team, really, both at Thermo and my team, without which none of this would be possible. I want to thank Mike and Vlad and all the Thermo group, and then particularly Jared Gefader, Phil Compton, Jack McGee, Raphael Milani, Ken Durbin, Brian Early, Ryan Fellers, and Louise Schachner. I also want to acknowledge the National Institutes of Health and funding our Proteomics Center, and the Keck Foundation and the Sherman Fairchild Foundation for its support. Thank you for your attention, and I'm happy to answer any questions. Thank you.
A stimulating presentation. I think we have a few questions in the chat box. Let me just open it up.
Sure.
Okay, so one of the questions is, for the individual ion approach, how broadly applicable is it in terms of molecular complexity of the analytes? In other words, how complex is too complex?
Right. Thanks, Jenny. And can you hear me okay?
Yes, I can. Yes.
Okay, perfect. Thank you. And thanks for the forum, and I hope you're doing well. Yeah, I made some claims there in the video, and I stand right by them. I mean, the complexity of the sample can be extreme, and we haven't yet found a sample where we can't generate a mass distribution, provided that you're in, the low to mid nanomolar regime. And as I said, these concentrations can be still quite low relative to normal micromolar type samples. And the complexity, you can have thousands, even tens of thousands of different molecular species in a sample and still get the mass distribution. And that was demonstrated directly in the talk. So I appreciate the question. And for those mass spec experts in there, let me just quickly explain why in 30 seconds. In the ensemble approach, in a Fourier transform mass spectrometer, the basis of signal is the constructive interference of the ions. Many thousands of ions, they have to compound on each other to give you this readout in the time domain. In the new method, it's no longer the basis of signal. You do not need constructive interference. It's individual ion. Each ion gives its own readout. And that's a little quick summary about why that is. So it doesn't matter that you don't have enough ions of the same proteoform, the exact same isotope log any. So that a long-winded answer, but there you go, Jen.
Thank you. So has your team performed any quantitative analysis? Did you find like the LOD or the lower limit of quantitation? for this methodology?
The linear working curve and its characterization. Yeah, just enough to know that, where things drop off in the, sub-nanomolar regime. We haven't done an exact characterization of the lower limit of quantitation. That'll require a bit more work, or other people can contribute to that with different analytes, would be different answers depending on their complexity. In the LOD, I'm comfortable with saying high picomolar range, but we need to lock that down on specific analytes.
Okay. So in one of the slides, you showed the calibration curve, the STORI calibration curve. You had one for denatured and one for native. Why is that?
Oh, right. Yeah, I guess it's a great question. I think I convoluted two issues on that slide. There were two instruments on that slide. So one was a Q-exactive plus and one was a UHMR. That's why. So each instrument needs a calibration. I would still want to cover the range of charge states over which you want to calibrate, and that in native can be quite different, quite much higher in charge. But in principle, you should need just one curve for one instrument.
Okay. Another question is I2MS compatible with chromatography, and any potential benefits?
That's so whoever asked that is intimating the future and got a lovely development vector for us mapped out there. So great question. Yeah, right now I would say that's really tough. And this is partly why the connection with sample stream, it makes so much sense. Sample stream allows you to elute in a chromatographic-like peak, but over a time frame that's at least a minute wide or 2 minutes, you can change it. You could have it be 5 minutes wide. And so if you have an especially complicated sample, chromatography with the detection on, say, a tribrid system or the Eclipse, it goes fast enough that you can do it. For I2MS, that's still a challenge, I would say. So you would need, a minute or two at the very least to generate some of these data. Now we're working on that exact topic, so stand by. Okay.
There's been a few questions regarding, is IMS2 feasible on all Orbitrap mass analyzers or instruments?
Yeah, so technically feasible, probably or sort of, where there's a lot of complexities to it, and individual ions are, very sensitive to things like the vacuum and why do individual ions die? There's many reasons. But most of the instruments we've looked at are Orbitraps that give 70, 80, 90% ion lifetime stability. The ions don't die up to 80. You need sort of 70, 80% of the ions not to die during this 1 or 2 second detection period. And not all instruments uniformly do that. So, but I think supported instruments like the UHMR moving forward into next year, that'll for sure be true. And so, I think that the rest is up to Thermo and Mike, Senko, Vlad Zabruskov, and all of their peers and coworkers to decide about that. But in principle, yeah, it's just, but some instruments could give low percentage ion survival rates. So that's something to look for.
Okay, thank you. So I have a question about, do you think I2MS will be a great real competitor for standard top-down approaches for complex samples comparative to multi-dimensional separations in the near future?
So I think the question is in the context of proteomics, top-down proteomics and discovery mode and trying to maximize the coverage. I hope so. I think it'll take, a few short years to make that a reality. And it's, of course, a question of how many resources we can funnel into the development. For now, I would think about it mostly as a targeted method for targeted proteoform discovery. Put it that way, where you've got an analyte, it might have thousands or tens of thousands of proteoforms, which is really complicated, but the tandem MS allows you to map PTMs. We've used it to do proteomics where we don't know the identity of the target. But for broad, large, high-throughput studies doing proteomics, that's not its wheelhouse right at the moment.
Mm-hmm. Okay. So going back to the top-down proteomics, in one of the slides you showed the sequence coverage of a protein using the traditional method and the tandem IM2MS method. And you also mentioned that we observe larger fragments. Can you explain why that is? Given that, I guess the fragmentation is the same.
It was exactly the same ions, exactly the same. Everything was the same. It's just the readout differed. Mm-hmm. And so it was an apples to apples comparison there in the data you saw for the TPI, the triose phosphate isomerase. And the bias difference between for the mass detection of the two are very different. The ensemble standard method advantages you to detect low mass B and Y ions. And for years I've thought, well, okay, so why are we not seeing the B300 ion, right? I mean, when's the last time you saw the B300, right? At a coffee shop last week. No, that was Elvis Presley. No, they just are of many charge states, many isotope peaks. They are there, but sort of in this rolling baseline. Now, switch to I2MS, and you get the very completely changed mass bias. You actually get better as you get higher in mass. So there's the B300. You can have coffee with him or her. You can just, there it is. And all of his friends, the readout is totally differently biased. I'm very excited about this because the sequence coverage goes way up, and the certainty with which you can localize PTMs goes way up. So I think when you think about other fragmentation methods on, say, the tribrid, and I just think it's a non-incremental gain. Sorry, Jenny, I get excited. And
it's all right.
I'm talking about Elvis Presley, right? Yeah.
Okay. So that's another question. What is the largest protein or protein complex that you've analyzed with this methodology?
20 megadaltons, in excess of 20 megadaltons. And so the size limit is, limited more about how you get the ions into the instrument. And so the UHMR is tuned to ions of even higher mass. So, I look forward to going to the 30 to 100 megadalton range. And the mass distributions can be generated so long as the ions make it to the trap, to the Orbitrap. And so I think that's the limitation.
Okay. We have kind of a two-part question. This I2MS technology, how do you establish a spray over many hours, like 72 hours? And how long does the computing time take to acquire this data and to deconvolute?
Yeah, so that's a great question. In the paper, the longest we've gone is 4 days. And that was to generate that one result on the 1,000 proteoform sample. But that was then, and this is now. And then, I mean, things are being dealt with. Like, hey, we were dealing with raw transients at the time, right? All these, very expensive in terms of compute, storage, and processing times, expensive. Now we don't have to deal with transients, and we're dealing with just much reduced versions of the data sets. So, it's already sped up tenfold. It'll speed up another tenfold. The processing time for both the file size, which is already shrunk by 100, and the processing times also will approach 100-fold reduction. And they were extensive. They were, it would take many hours to process such a data set. But now we're down to an hour, and for even a very long, direct infusion experiment. And to answer the second part of that question, it was, just simple syringe pumping for days into the instrument and reload a syringe. But this is why the sample stream, you can't do that with a nanogram of protein, right? So the sample stream piece of this is to be able to take tens of nanograms, change their buffer, control access to the sample with high efficiency, introduce it for say 5, 10, 30 minutes, 70 minutes. And that's what we've got going with the sample, the combination of the two.
Okay. And then I think we have enough time for one more question. Is the ITMS technology, including the software, already available on Orbitrap platforms, including, for example, the customer has a Q-Exactive biopharma instrument?
Yeah, that would be a great instrument to focus on. And, I would refer to Vlad Zabruskov and Mike Sanko on this one, or defer to them. But yeah, technically, yes, it's through a beta test program. And so, go chase those two folks down, or the folks at Thermo can connect you up with the right folks and emails, and get into that program. I think it'll be, a few months more before they can expand access, but certainly by, next year, and the timelines up to Thermo Fisher Scientific. But yeah, so it'll be available shortly in a more widely accessible format, and the UHMR is the main focus point. So the biopharma is, I guess the technical point. Let me just address the technical question, and that is that for native ions, the center electrode voltage is not a problem. Where you could have a 5 kV center electrode or a 1 kV, and things basically function well. Good ion survival rates. For denatured mode, especially when you get higher in mass, you need to reduce the voltage on the center electrode. And so that's just one type of technical consideration. The types of ions differ in their survival rates depending on the center electrode of the Orbitrap. So at the center electrode voltage. So yeah, I hope that answered basically the question, Jenny. And I just want to thank Julian Saba and Claudia and all of the collaboration partners at Thermo Fisher Scientific. And for all the listeners, thanks for chiming in there. And we look forward to 2021.
Thank you, Professor Kelleher. That's a very good presentation. And thank you to the audience for listening and participating. So we will reconvene in 30 minutes for session 2. So with that, I'd just like to close the session, this session anyway. And I'll speak to you and see you in half an hour. Thank you. Bye-bye.